So, hello everybody, welcome to our webinar on state-of-the-art reaction monitoring and parallel synthesis. My name is Martin Winter. I'm the founder and managing director of Lab Automation Network. I'm calling in from Tübingen in Germany, and it's nice to have you all here in our webinar. Our webinar series as a whole will be focusing this year on special and outstanding technologies in the field of laboratory automation and digitization. We will be having interesting partners and companies presenting their technologies. There will be technologies on semantic or webinars on semantic technologies. There will be a webinar on digital liquids, so digitization of liquid samples and many more. And for today, we will be focused on parallel synthesis and reaction monitoring. Just as a quick view on the agenda, what we'll be having today. First of all, I will give a quick introduction to Lab Automation Network, about what we are doing and who we are. Then I will hand over to Mark Spears from Virscidian, our network partner who is offering solutions for chromatography and LC-MS data analysis, which is really outstanding in the market. Then Mark will give you a live demo and show how Analytical Studio is able to perform high throughput reaction monitoring. And then in the next part, we will switch to another partner, the second company for today, which is Nevolab. Nevolab is producing reaction blocks for parallel synthesis, not only for process development, but also for other applications, because it's also important to perform those syntheses that we want to monitor. Yeah? So the second part is a more practical part. And Manfred, one of the managing directors of Nevolab, and Matthias will show how these reaction blocks really work. And then at the end of the session today, we will have 10 to 15 minutes of Q&A. So please feel free to ask your questions, put them in the Q&A section, and we will answer them maybe live, or if there are too many, also after the webinar, we will contact you and answer your questions. Please make sure that you acknowledge that this session will be recorded, so that you can see everything today, and we will then make this session available after today for more interested partners or customers. All right. As a quick introduction to the people involved today, Mark Spears is a chemist, analytical chemist. And yeah, he is working for Virscidian for quite some time now, has a broad range of experience within pharma industry, and is very passionate about improving quality and efficiency in high throughput workflows. Manfred from Nevolab, who will take over for the second part, is also a chemist and has been working in the lab for many years, and is also active in the field of lab automation and laboratory technologies for many, many years. I've worked with him, let's say, more than 15 years, for more than 50 years now. And I'm also a chemist and in lab automation for more than 20 years now. So what about the Lab Automation Network in general? We are an alliance of technology providers and solution providers in the field of lab automation and digitization. And our business model is to select the technology leaders in their specific domain and bring them together as a network to perform engineering projects and development projects, and to provide our end customers in pharma, chemistry, and biotech with outstanding lab automation and digitization solutions. And that does not necessarily involve robots. what we will see today is another example with very simple, or let's say very small equipment. You can also do a lot in high throughput technologies, and on the robots we will focus in later webinars. So these are the applications that we typically work with, and first step here is synthesis and formulation, and reaction monitoring is part of that. The technologies we use is not only robotics, but also laboratory instrumentation, software, analytical technologies, and many more. All right, so let's step into the topic of today, reaction monitoring and parallel synthesis. And our first speaker for today is Mark Spears, and I'd like to hand over to Mark now. Please, Mark, the stage is yours.
Thank you so much, Martin. I will go ahead and share my screen as well. And I want to say a quick word of thanks to everyone for tuning in. We really appreciate you taking time out of your day to learn about what we do and how we are impacting data analysis in the pharmaceutical industry. So I want to give you a quick introduction to who Virscidian is. We were founded in 2008, and we were founded out of years of experience coming up against roadblocks that scientists were experiencing in their day-to-day work with data and their experiments. And so we were encountering, the founders of Virscidian were encountering these roadblocks, and became very passionate about helping scientists to overcome these problems and provide software solutions that offer real benefits in quality and efficiency. And so Virscidian is still passionate about those same exact things. And we are on a mission to deliver software solutions that overcome the challenges that you as a scientist experience in your lab every day. And the ways that we do that is we really try to take what you as a scientist do in your natural workflow in your laboratory and the way that you think about your data. And we try to then put that into a software solution and offer an end-to-end analytical software suite that tackles those challenges. Now, I'm going to be showing you our desktop software today, which is called Analytical Studio. But I also just want to make you aware that we do provide enterprise products as well, which are able to automatically sweep data from instruments, automatically process them on a server, and provide those data or access to those data anywhere that you have a connection to that server. So we do offer desktop and enterprise products. And as Martin pointed out earlier, we are offering world-class chromatography and mass spec data analysis, processing, and reporting of results. Now, Virscidian is offering many, many different workflows today. We were initially in 2008, when we started, we were offering compound QC. And as you can see, that has expanded quite dramatically to many different workflows in the medchem and drug discovery space. So we're doing calibration and quantitation type workflows. We're, of course, doing synthesis, reaction screening, reaction monitoring. That's what we'll be talking about a little bit later. And I'll show you a live demo of some of those data. We're also doing chiral and achiral purification, where Virscidian has really become a market leader in that workflow. We also offer instrument suitability. So this is down here in the red space. This is a module that we offer, which is able to track the performance of your instrument, automatically provide alerts if your instrument is trending out of spec or out of calibration. And then we also have now become a market leader in the affinity screen and mass selection space, so ASMS, or maybe you have heard of it as ALICE in your laboratory. And now Virscidian is also branching into several other areas of interest. So, for example, protein and deconvolution workflows, whether that's DNA encoded libraries, oligonucleotides, or any sort of large multi-charge state type of molecule. And last, I just want to mention that we are working on a synthesis design tool as well. So this is a tool which will allow you to design the types of experiments that you see today. That is connecting to an internal library of chemicals, choosing the chemicals that are involved in your screen, laying those out on a plate, providing sample lists and stock solutions pages and all the kinds of things that you as a scientist need to do to get ready for and design a synthesis screen, like we will see in just a few minutes. So we're very busy people. We have a lot going on. And I would love to chat with you if you're interested in any more information about any of these topics here. Okay, but without further ado, I really want to get into Analytical Studio. And so now we will switch over to a live demonstration of our software. Okay. So this is Analytical Studio, and I want to introduce you to all the different parts that you're seeing here on the screen, and we'll touch on each one of these. It may look a little bit overwhelming at first, but we'll touch on each one of these different panels and try to give you an overview. And I do want to just say what I'm trying to do in about 15 minutes today. We normally take about an hour to present, so you'll have to hold on tight and keep up. We'll move somewhat quickly through the different aspects of the software. So of course, we as scientists understand that high throughput experimentation and reaction monitoring is not necessarily a simple thing. There are many different types of approaches to these experiments. So for example, you may have a different reaction in every vessel or in every well of a plate with a different desired product in each case, or you may be after the same product in all wells, but just vary some sort of reaction condition across the plate or across different vessels. For example, different solvents, temperatures, pressures, concentrations, et cetera, et cetera. Additionally, you may be doing time course or reaction monitoring types of experiments. That is, you're taking samples at various intervals as the reaction proceeds, and you are monitoring the change of an abundance of different products or substances or impurities or something like that over the course of time. So all of that to say that whatever solution we provide for HTE needs to be very flexible, and it needs to allow the scientists to mimic the way that they work and get out the results that they need for their experiment. And so that's what we've tried to do here. Now, Analytical Studio is very substance driven. And what do I mean by that? I mean, first of all, let's focus in on this panel in the bottom middle of your screen. This is our substance editor or substance list here. So Analytical Studio is able to read either from the raw file or from some other source a list of substances that should be identified in the sample. So in this case, as an example, we have our product, we have an internal standard, we have a known byproduct, we have a known starting material, and things like that. Of course, in HTE, sometimes we know a list of impurities and byproducts and side products and catalysts and all kinds of things that are involved in the reaction. Sometimes we don't know what to expect. So we could have either of those cases. Where we do know what to expect, we're able to then look at this product. We know the formula. We can then look at the underlying masses in this peak as an example. And we can see here that we are indeed assigning the product based on an isotopic pattern match. And if I zoom in here, of course, we have the M plus H, we have the M plus sodium, and we can search for several other adducts as well to confirm that this mass is indeed found. And then we color the peak according to whichever color you have chosen in your table here. This is a nice green color for the product. Now, we are also able to do things like color this plate according to all of the places where this product is found. So for example, all of the wells here are green because I've found product in all of the wells. If I go to the internal standard, it's sort of a pink color. And you can see that indeed we're finding the internal standard in all 96 wells, which is great. That's what we would hope to find. And we can do the same for all of these other substances as well. Now, this is great if you know what the substances are that you're expecting. However, in HDE, we often find that we're producing peaks that we don't know what they are, right? These are impurities or things that we did not expect. These are unknowns. And so how do we handle that? Well, in Analytical Studio, we have the concept of auto assignment. So that is to say that we take peaks which are not automatically named as a known substance, and then we name them, we give them a name and identify them. So in this case, all we're doing is identifying them by a relative retention time. And this is all relative to the internal standard, of course. So you can see here, these peaks in the table are listed as simply 0.74, 0.75, 0.9, and so forth. This is very useful for identifying all of the unknown peaks above a certain threshold percentage, area percentage of the chromatogram. And then this enables us as well to look across all of the different samples in the plate or in the reaction, different reaction conditions, and say, how many times does this particular impurity at this particular retention time occur across all of these samples? And indeed, we have a panel here called Global Substances, which allows us, pardon me, to look across all of these samples and say, for this particular named substance, how many times does it occur? And so here we go for the internal standard. It occurs in all 96 wells. Same for the product, all 96 wells. But then the starting material, it only occurs in 67 wells. So in many cases, we're consuming our starting material. That's great news for us. That means our reaction is going well. And then you can work down the list. Here we go. Here we're looking at something at 0.9 minutes. And this peak is an unknown, but it occurs in 53 out of 96 wells. So clearly something that is occurring with this frequency across these samples is something we need to pay attention to. And so you might be able to then focus in on this particular peak, look at the mass which is present. So here, of course, it might be whatever this is at 454 Daltons. And then you can, with your knowledge of the chemistry of the reaction, you can make some determination about the formula of this particular peak. All right. I should say as well, when I click on this, you're able now to see all of the places that occurs across the plate. So now that we have our peaks assigned, we're in a very good position to start understanding these results in more depth. And of course, you may want to perform some different calculations based on these results. That's very common. And so in Analytical Studio, we call these calculations expressions. And so that's what I'm showing here again on the bottom left-hand corner of your screen. And expressions are not only customizable calculations that we can do on the peak characteristics, but we can also use expressions to report out many other things about the sample. So for example, what is the conversion in each one of these conditions? What is the area of each substance? What is the number of integrated peaks in this chromatogram? What is the relative ratio of each substance to an internal standard? Or you could even do things like provide warnings for samples or wells which have some sort of problem. Maybe it's a very weak UV response, or some other, or your conversion falls below a threshold, let's say, something like that. So expressions are very flexible and customizable to what you want to get out of the data. And we can, this is great. We can export these results in a like an Excel table or a PDF. But we can also interact with these things directly in Analytical Studio. So for example, if I now color the plate by the selected expression, and I've selected conversion here, I can now see in a heat map type of arrangement, which wells had the highest conversion, which had the lowest. And here I'm looking at a color spectrum from green, which is the highest conversion, down to red, which is the lowest. And so you can very quickly get an overview of the plate and how each one of these samples performed. But I do want to say that even though this is excellent, we're starting to get a better idea of the results. I do want to say that this is only one dimension of the results. We're only seeing one dimension of how the reactions performed, but it really doesn't tell the entire story. What if we want to include many aspects or many dimensions about these results and have that inform our decision? Because after all, in HTE, we don't simply care about maximizing the product. We also care about things like minimizing other impurities and minimizing other side products. We also care about things like what is the relative amount of known substances compared to unknown substances. And so how do we bring all of that information together and then make a good decision about which reaction condition is best? And to do that, we have charting tools built into Analytical Studio. And I want to just quickly walk you through this chart as a nice example of what is possible. On the x-axis, we're plotting the number of peaks. This is the number of integrated peaks in a chromatogram. And as you might expect, a lower number of peaks is better. So for example, if I click on a sample here that has a small number of peaks, I see a relatively simple chromatogram. If I click on one here on this other side that has many, many peaks, I see quite a complex chromatogram with many, many different colors and integrated peaks along this time. So clearly for us as HTE scientists, a smaller number of peaks is better. And it means a simpler result, and it means that we are not producing impurities. So we would choose something on this left-hand side of the graph. Now, on the y-axis, we are plotting conversion percentage. And so clearly a higher conversion is better. So then we would be focusing in on something toward the top of this graph. So immediately you can see that we are narrowing in on the top left corner of this plot. However, there are two additional dimensions to these data. Number one, we've colored these results according to conversion as well, and we put them in buckets or thresholds, if you will. So green is 100%, and then the light green is a little bit less, and then yellow and so on down to the orange color. We have also changed the size of some of these dots. And the point size here corresponds to the relative area of the chromatogram, which is assigned to known substances versus unknown. So in this case, a larger dot would be desirable because we would have more confidence in that the substances which are assigned are known substances rather than unknown impurities which were produced. So we would want a larger dot. And so really what we're plotting here is 4 dimensions of the data. And you're able then to narrow in on this upper left-hand corner in the large dots. And these would, of course, be your best results here, these couple reactions. So I hope that makes sense to everyone. You're able to follow these charting tools built into Analytical Studio. Now, there is one more important feature of a good reaction condition, and that is a product peak which is well resolved from other unknowns or even knowns surrounding it. And that, of course, means that this reaction can more easily be carried forward into other stages of research. So in Analytical Studio, you're not only getting the result of which reaction condition is best, but we can go one step further and we can even predict which gradient method will be the most suitable for carrying forward into other stages of research. And this really sets Analytical Studio apart from other software solutions that are on the market. So if you have been someone who is sitting at home wondering, what are these green and red bars on this chromatogram? Well, now I can reveal to you their purpose finally. So what we have done is inside of our processing method, we have included a lookup table of different gradient methods that are available on the instrument. And typically speaking, this would be reserved for purification workflows. But it's also very applicable for HTE as well, because we can more knowledgeably choose a reaction condition and then send that out to someone so they can select an appropriate gradient method. So what we do is based on the retention time of a product peak or multiple peaks. That's also, we can also handle multiple product peaks in the case of isomers. Based on this retention time, we select the appropriate gradient. And of course, we have a start and an end here. And, and then we overlay this on the chromatogram, the start and end times, and also the name of this particular gradient that is selected. Now, we always allow the user to override this choice if they so choose. However, our software does a great job of selecting a gradient, and many in the purification and HT workflows rely on this, on Analytical Studio to make this choice for them. This gradient method can, of course, be exported along with other results so that it can inform later stages of research for you. All right, so now we have a really nice picture of how these reactions performed. We can narrow in on which conditions are best based on our knowledge of this layout. And of course, we need to report our results. So we can do something as simple as reporting an Excel batch report. So if I go to File, Export, and I'll just do this in real time for you here. I'm going to do an Excel output and send it to Excel. We can also send this to a Spotfire if anyone is interested in that. We can create a Spotfire readable file, which you can just simply import, and then you have access to some additional charting capabilities and graphing capabilities. But I'm just going to simply do Excel for now for all samples, and I'll export that. I'll overwrite the one that exists now. And there we go. So this would simply be just be a simple example of the types of Excel reports that you can generate. Here are the names of the samples, acquisition date, the MS method that was used to create the sample in the first place, its location, and then some key results for that particular reaction, conversion, and number of peaks, and so on. Okay, so I hope that gives you a nice overview of Analytical Studio. We could imagine doing the same thing for time points as we could for reaction conditions. And so there's a lot of flexibility built in here. And we've also put a lot of effort in trying to help you very quickly narrow down to the most important results and the best results, even though you are considering a large number of samples. So I hope that gives you a nice overview. And with that, I would like to turn it back over to Martin.
Thank you very much, Mark. That was really impressive. And I think Analytical Studio is really a very, very powerful tool to look deeply into what's happening in the reactions. Yeah. So, yeah, but where are the samples coming from, right? Maybe some of the participants have robotic systems for parallel synthesis already, but you don't need something like that. You can do this manually, and one very efficient way to produce your samples in multiple parallel experiments will be shown by Manfred and Matthias from Nevolab now. So please, Manfred, the stage is yours.
So yes, thanks for joining our webinar here. Hello to everybody, and thanks Martin and Mark for the first presentation here. And I would like to introduce here the company a little bit, that you know what is a company about. And later on, I would like to have a two-part presentation. One part is more general about the prerequisites of a reaction, and the second part will be more practical out of our application lab done by my colleague Matthias in our facility in Mayerhöfen near the Lake of Constance. So what is Nevolab about? So we have nearly 20 years of experience in products and service focused for people working in labs. So this is our focus in general. Our background is we have produced some OEM products for microwave synthesis, and also did a lot of things in sample preparation, and also in customized solutions for our clients for special questions or some requirements in high throughput analysis and high throughput synthesis. And what we always looking is to also integrate lab data management tools into the devices and the products to make it more accessible and more feasible for our customers. In general, for our perspective in company, so we created some products, and one of them is our XELSIUS reactor block, and also an automated system. We call it XELSIUS STS, means sample solubility test and sampling. And also we're creating some smaller tools like a syringe pump, or here a solvent control tool, which is used for HPLC users to provide solvents and collect waste out of high throughput HPLS systems. Today we will talk about reaction monitoring. And so the idea for my presentation, of our presentation, is what is important for your experiment for reliable data, and what is a prerequisite for you to create reliable experiments that you can take later on for your results. Okay, I'm sorry. My colleague showed me here that my presentation is not be shown. I will share my screen. I'm sorry for this. So hopefully everybody can see my screen now. So part one is focused on an instrument, or what is important from the instruments to create reliable data. And I will tell a little bit about reaction parameters and of the sample parameters. And the second part will be done by my colleague and shows our reaction block itself and give you an idea what we did to provide.
So Manfred, just we can still not see your slides. Now it's coming. Now it's coming. I hope everybody can see.
Sorry for this.
Yeah.
Can you see now? And
I can see it. I hope everybody else too.
Okay, part one is about general, and part two will be done by colleague in the application lab and show how the instrument works and what we did to produce high quality data on high quality experiments for your data to find out what is worth to work over and go further. So the most important thing for reactions and experiments is reproducibility. And in a reaction, so we have maybe a couple of parameters, and most important thing is temperature. The second one is concentration, and the solvent and the stirring is important. And for some reactions, the atmosphere and the inerting is important. And when you're taking samples for the analysis, so the sample taking process is also very important, because the errors in the sample taking cannot be compensated in the analytical method itself. So everything is focused on reproducibility of everything, and starts with the reaction itself. So when we talk about temperature, we have here in our instrument 10 different reaction cells, and here gives an idea about the checkage temperature, how important it is to have the same temperature management in each reaction vessel. And it's a question between the vessels, but it's also a question of the time. So you need to rely on, when you start a reaction in an instrument, let's say on Monday, that on Friday or some months later, the same performance of the heating program will be performed. And also important is, when you compare different setups in terms of vessels or in terms of solvents, that also this reaction setups will be comparable for reliable data. And here I will give you a small idea what is the influence maybe of the reaction vessel, and the dimensions and the tolerances and the wall thickness of the reaction vessels. So we tested a couple of reaction vessels and created an easy cooling down process. And you can see here that the end result of the temperature, so we try to have -10 as our target temperature. You see in some vessels we can achieve -10 or -9.8, but also with some thicker and more tolerances in a reaction vessel, we only achieve 8 degrees. So it's important for you to know if you run your experiment at -10 or at -8. So this is important to have a reliable temperature management and have a good accuracy in your reaction vessel. And another question, especially for long-term reactions, is evaporation of the solvent. So when doing long-term reactions over, let's say, 12 or 24 hours reactions, and you boil it at a higher of the boiling point of your solvent, and you have maybe not a closed vessel, so there is a loss of solvent to observe. And for this, we checked here a couple of different solvents, and we boiled it for 24 hours, and one with a reflux condenser shown here on the right side, and on the side without a reflux condenser. And to make sure that some solvent could be evaporated, we put a syringe in, and so that the system is still open. And here you can see the results, maybe here for ethyl acetate. So we boiled it 10 degrees higher than the boiling point, and you see the dark blue after 8 hours. From 10 milliliters, only 8 milliliters will be left, and after 24 hours, maybe 1 or 2 milliliters will be left. And with the reflux condenser, you see you have a much better recovery of the solvent in your reaction vessel. So we ended nearly up at 9 ml out of 10. And with acetonitril is a similar one, and you see here with the reflux condenser, there's nearly no loss of solvent, and without, we have a loss maybe of 5 or 6 ml after 1 day cooking. And with let me see here with low boiling substances, so the effect is much more important. So with this acetone, so we have after 4 hours, maybe even with reflux condensers, a loss of maybe ½ ml, and come up to 8 ml at 1 day. And without a reflux condenser, you see we will have maybe a dry solution after 24 hours. With methanol, it's a similar one, but it's not as worse as it is with acetone. When we focus the inerting atmosphere, so we choose the oxygen-sensitive reaction like a Buchwald-Hartwig amidation, and here it's very important that you have a good how to call it, a good yield for your, or a conversion rate for your reaction. And so this was the standard for this reaction shown here. So the conversion rate or the result of this reaction ended up nearly with 80% of the tests. And so we developed with our customer an inerting unit to make it easier to evaporate the atmosphere into the reaction vessels and do it in a serial way, so that we have the same conditions in each reaction vessel. And you see with this process 10 times vacuum and argon and 3 seconds of cycle. So we ended up at, let's say, between 90 and 94% of the results. And when you do it with 4 times vacuum with 60 seconds of each step, so you come up with a very, very good result, higher than 94% in general. So you can see with some setup and some tools, you can improve your yields only by the instruments and the accessories you are using for your reactions. So we have the reflux condenser here, we have some inerting units here, and also important is to have some caps for sampling and bring some solvents into reaction in an easy way. And the second part is, if your reaction is still fine, it's also important to think about the sampling of your reaction for the later analysis. And for this, you see, we have the components, and also the concentration and time is important. And so sampling is the time when you take the sample is important for your reaction kinetics. And also important is the concentration and the dilution later on. And you see, we took here some experiments for our instrument, and we have chosen some slurry systems. Here we use titanium dioxide slurries to show how good is our dilution and sample taking process out of our reactions here. And we collect it in some HPLC vessels for later on analysis. And you see here the result. And we also did it with some different solvents and different viscosities of solvents. So we have chosen water sugar solutions with 41 bricks. It means we have 40 gram sugar in 60 gram water. So when we tested it with 200 microliters sampling out of our reaction vessels, and you see the reproducibility was excellent. We have less than 1% in a reproducibility, and in terms of accuracy, we checked it with the density. So we came up with an excellent accuracy, lower than 5% in total. So the only thing we have a little bit problem is here the water oil system. So we have a 2-phase system, and for this we forgot to switch the stirrer off. So this 12% accuracy is a question of an error in the sample taking. And the same result is, or a little bit worse. Reproducibly is done when we take only 50 microliters out of our reactor. So we came up in a reproducibility less than 4%. And also the accuracy is in the same range than we have with the 200 microliters. So this is my overview about the importance of the instrument for your data and the reliability of your data. And now I would like to switch over to my colleague to give you an idea how this machine looks like and what is the performance and the idea out of our application lab.
Okay, so then thanks Manfred and hello everyone. And I will start sharing the video. This button, okay. So, the XELSIUS device. As you can see here now, we have a standard XELSIUS device. I'm not sure if everyone is highlighted. I can see Manfred only now. Do you see my screen right now, my camera? Or is it not highlighted?
Yes, I think we can see it. Thank you.
Okay, okay, good. So, okay, then let's give a very quick overview about the device itself. We have a touch-sensitive monitor to show you the state of every cell every time, and of course work in the lab even with gloves on if you want to. We have our power supply unit, which proves, of course, the power for the XELSIUS reactor and has an internal Windows 10 PC, so have all the benefits of it, data export, network communication, and so on. And we have our standard XELSIUS reactor with 10 completely individually cells. So this means if you want to heat up in a one cell, then the cell next to it is not affected. You can same time heat and cool in different cells if you want to. Next of it is the reflux condenser. And like every tool of us, it's connected by non-spill coupling. So there's you do not need to be afraid to lose some water, for example. And you can just put it onto the reactor and connect it if you want to. You can connect it in line with our device with this little tube, and then just place the output of the external chiller. For example, just that's one of those guys to use one water circuit. Of course, because of the design, you also can use another liquid circuit if you want to use a high temperature difference. So then next part here is the, let's have a closer look for a better view. So next part here is the inertization unit, which can be used with our with our vials to create oxygen-free reactions. So you can evaporate the atmospheres here, or for example, use another atmosphere like nitrogen if you want to, and then just place it into the reactor en bloc after you prepared it. And we also have 3 types of vials we provide. There's an PTFE high volume vial. There is an high volume glass vial, and now I need more fingers. There also is a low volume vial. So if you want to use with reduced volumes, reduced sample amounts, then this is your vial to go. It's a little bit smaller here. And if you want to use them, there also is, of course, an adapter which can be used to optimize the temperature transfer from the standard reactor to the low volume vessels. So yeah, so much about a very, very quick overview about the standard accessories of the XELSIUS reactor. So next I'd like to show you is a little bit about the software and how you use the device. So so therefore I'll connect to the reactor via VNC. To give you a better view than the camera. So okay, now you should see the screen. So you can see here now the XELSIUS lab mode. It's our software to provide an as fast and as easy as possible way to do your reactions and to adjust this device. So as I said, each cell is completely individual. And this also means, of course, each cell can be controlled completely individual. So we can see here the cell is gray, which means it's not active right now. So therefore I can click on it. And here I see all the parameters possible for the cell. A target temperature, I can click on it, of course, to change it. For example, let's change it to 100 degrees. I can define, do I want to reach my target temperature as fast as possible, or do I want to use a ramp in Celsius per minute, or for example, a crystallization reach the target temperature very, very slow in Celsius per hour. I can define the stirring speed, and I can activate a timer. So if I want to cook, for example, my sample for 5 hours, and then I want to shut the system down by itself, I can click on the timer, can click on the numpad here on the bottom, and then I can see here now I want to reach 100 degrees. I want to do it as fast as possible, stir it, and after 5 hours, shut it down. And to reduce the amount of clicking, of course, you can now say, I do not just want to edit one cell, I want to apply the settings to the whole left side, or I want to remove the settings from the whole left side. So you can save a lot of time and clicking errors by parameterizing them just once. Okay, so let's say 3 cells, 100 degrees. I click on OK, and we can see here now the settings have been applied. And if I click here on the circle symbol, you see it will switch to an error, and the cell will move to green state, which means it's active. And you can see here now we are heating pretty fast. And same time, we can click on another cell, which is not in use, and can define a temperature, let's say 0 degrees, apply the 3 cells just connected to them, start them same time. As you can see here, there's a little difference between using timers and no timers. If there's no timer defined, it will count upward to show you how long the cell is active. And if you use a timer, of course, the opposite is the fact. It will count downward to 0 and then deactivate it by itself. So you can see here now we are about a minute, a little bit more than a minute, and we already reached the fine adjusting part, which means the cells are not powered full power. They are trying to reach the target temperature very, very accurate and fast. And same time here, we cooled down in after an hour, 20 degrees. So we are here very quick. Same time, we do it very stable and reproducible. Okay. Then next thing in our software integrated here is the catalog mode. So if you want to record the data, to show them and trace them, there is the catalog mode for, and to switch between the modes, just click on this little temperature probe symbol here to switch between the set point mode and the profile mode. And if I now click on the cell, you can see here, we can define here a multiple step method. So we can define a start temperature and a holding temperature and an end temperature, and every part here is time controlled. So we can say, reach the first temperature for 4 minutes, heat down over a ramp of 2 minutes to 72 degrees, and cycle down after that again over 3 minutes and hold the target for 3 minutes. And here, of course, we have the option to cycle those procedure so that we have the option to redo this whole method here 10 times in a row or up to 150 times if we wanted to. Integrated in this profile is the catalog, a local database to save or restore your settings. And if we open it, you can see here the database, which allows you to, for example, here, click on an empty place and click on apply to catalog. Then your profile you just created is saved. So next day, if you want to, you can just click on it and click on apply to profile. Then it will be imported again. And of course, in this mode too, it is possible to parameterize multiple cells same time if you want to, just by adding and removing them. And if I now click on the cell and activate them, then you can see here the biggest change in visual, the graphic display of the temperature history. So if we click on this zoom button, we can see here in details the temperature profile we want to reach, each step colored in its individual color, of course. We can see the actual temperature of the cell, which is also, of course, here in this red line displayed and recorded the whole time. And if there is an external temperature probe, for example, there's one connector for 50,000 on the back of the reactor, so 1 50,000 each cell is possible. It will be also here recorded and shown same time. And you can, if you want to, of course, you have the full-time possibility to zoom in and see in detail how your reaction was processed. You can zoom out and modify the visual as you want to. And here also you have the options to export the data by as a PDF, and of course as an Excel file or a CSV file if you want to save them.
So thank you very much, Matthias. We are running out of time. Now we are nearing the end of our session today.
No problem. That was just a finished part.
Yeah, that was really impressive. So, and I think everybody has seen that it's not just a reaction block. It's also equipped with a small liquid handling arm with the functionality to take samples. And the samples can then be transported manually or in an integrated robotic system to HPLC and analytical instruments, and then produce the data that later on will be processed by Virscidian's Analytical Studios. So for the Q&A session, please Mark and Manfred join me for answering some questions. I think we can do this also by email. If you have any more questions, write us an email. We had one question from one of the participants regarding the resolution of the mass spec functionality. Is it at a resolution that can estimate isotopic distributions and predict the number of carbons? That is a question for you, Mark.
Yes, the answer is yes. So if we are working with high resolution data, we are actually able to estimate the number of carbons and potentially even provide the chemical formula, the empirical chemical formula of the unknown substance. Exactly.
Perfect.
Mm-hmm.
Okay.
That functionality is built directly into AS Pro as well. I did not show that today, but that is possible.
Very good. All right. So any more questions will be answered by email. Thank you, everybody, for joining. Thank you to all the speakers and presenters of today. And as a final message for now, we will have our next webinar on 15th of July with a completely different application, which is single cell injection in neuroscience. So injecting solutions like CRISPR-Cas or others into single neurons. So also a very interesting topic. Thanks again for joining. Thanks and have a nice day and hope to see you soon at our next webinar. Bye-bye.